We're going to start with the first laboratory science question, and that is, what are the most important mechanistic questions related to traumatic brain injury that remain unanswered? And we'll start with volunteers.
I'll start. So I think everybody in this room has a favorite mechanism that they like. We all have our hand on a different part of the elephant. And to me, there's an overarching mechanistic question that's related to TBI that remains unanswered. And we've touched on it in a number of these presentations. It's really the when and who to treat, and not just what to treat with. And by the when, I mean, what are the presenting conditions for which you would use this therapy? So the mechanisms are, we all think of those as leading to therapeutic interventions. So the when to treat, who is it that we're supposed to be treating, and what time, and for how long should we be treating? Those to me are really important, the when and the who, not just the what mechanism.
I agree, Susan, but the central issue is that it has to be reframed in the context of trauma. Because
yes.
Many of us are speaking about mild traumatic brain injury versus the more severe end of the spectrum. So I think as we go through this and talk about these laboratory issues, I want them framed in the context of mild versus severe TBI. Mm-hmm. So I think that's an important question. And that also goes into the model development, which is part 2. But I think we've heard already from our experience with pigs, and Dr. Kilball, is that correct, from your group? I thought fascinating work on pigs, is how we're going to, and the work we're doing in rodents, how we translate this to the human and the when issue. And I don't think that Dave Hovda posed that question. I do not think we really answered that. And frankly, I don't think we have used that, answered that. And I'll get back to question 1, but I think it's a very important issue. You're doing biomarkers, we're doing biomarkers, OBTT's doing biomarkers in rats. Many of you, and I know Professor Ramon Diaz-Aristide is doing biomarker discovery. We really need to correlate all these laboratory findings to see if the rat and the pig and the human move in tandem, and I don't think they do. And that also could then influence the what and the when of this mechanistic question. So I'd like people to think about that, because clearly I have had, unfortunately, in my early in my career, did a lot of human pathology. And I will tell you, the human response to trauma, DAI, is completely dissimilar from what I see in rats, but much more pig-like. I don't have all the data to publish that definitive paper, but it poses a lot of mechanistic issues. So I somewhat digressed from the question, but I wanted to frame it again, mild, severe, and also modeling issues as we move forward.
John, I wanted to ask you, in relation to comparing diffuse axonal injury in the rodent versus the pig, for instance, are you talking about a difference in the distribution or a difference in the time course of development of that axonal injury?
One, as the gentleman alluded to, is the magnitude. I mean, obviously these are gyrocephalic animals, so literally you could touch the pig brain and you see damaged axons. So the magnitude of the response is stunning, and the temporal progression, in my estimation, is much slower in humans and pigs than rodents. We need to confirm that, and we need the OBTT and other mechanisms, and hopefully the NIH will help us as we move forward. But I think those are very key questions that need to be addressed, because they drive therapy.
I just wanted to comment, John, you and I we've talked about this a little bit before. I tend to be on the other side in terms of the comparison of the therapeutic window between the rodent and the higher species, including humans. I don't think we have enough data to conclude that they're different windows, but I bet that I don't have enough data to prove that they are the same window yet. But that is a critically important question, because if you look at interfering with particular mechanisms of secondary injury, you've got to understand in your target species what's the duration of that, because obviously you would intuitively treat with drug X, Y, or Z for a sufficient time to interrupt the elevation of that mechanism. Another point I would make in general, irregardless of what your favorite mechanism is, that when you go to finding a magic bullet off the shelf or creating one, you've got to really do careful pharmacological experiments that are very complete, and they're very onerous in terms of their difficulty. You've got to do target mechanism demonstration. You've got to demonstrate that your drug affects your mechanism of interest. That requires a lot of laborious dose response. It requires looking at defining the optimum duration of treatment. Obviously, therapeutic window, and again, we need more work to determine the comparison of therapeutic windows. Also, we need, very importantly, this is another feeling that I have, and based upon a lot of data, that if you define the blood level, or the brain level ideally, but say the blood level of drug associated with your neuroprotective effects in your animal model, at least that gives you some basis for translating it into humans. It's hard for me as a pharmacologist to believe that the optimum dose level the optimum concentration of the drug in the plasma associated with neuroprotection would be that different between animals and man.
So to answer the question about important mechanisms, I agree with what everyone said. I think neuroinflammation is probably one of the most important targets that we need to think about. We now know that there's good and bad inflammation. We think inflammation may have an important role in the progressive atrophy that we see and increased probability of neurodegenerative diseases. So I think in terms of, again, using biomarkers to look at neuroinflammation and new strategies to manipulate M1 and M2 macrophages, microglia, and things like that, I think that's something that's going to be extremely important in terms of the TBI field. But there's a lot of mechanistic questions that we think about every day, but this is something that's very timely right now. And I think the science is getting a little bit clearer in terms of inflammation.
But Dalton, following up on that, and I think I agree with you, but Dr. Kilball noticed it, and Dr. Lafreniere noted that the inflammatory response in rodents is quite dissimilar from that seen in higher order animals in man. So again, this is a classic example. I agree with you, but we have to be very careful as you move forward in discovery.
I'd like to echo what Dr. Kirsch said this morning about looking at mechanisms of spontaneous recovery. It is a huge amount of spontaneous recovery that we don't know why our rats get so much better in just a few weeks after injury, and humans do much better over 6 months after injury. Conversely, there's an amazing amount of reserve, so they can have underlying deficits that are masked by their ability to have this reserve. For example, Parkinson's, you can lose 90% of your dopaminergic neurons before you show deficits. So there's probably more damage than we're aware of, and it's being somewhat compensated for.
Yeah, I would echo what both John and Doctors Dixon and Dietrich have said. But teleologically speaking, if we couldn't recover, if we didn't spontaneously recover from traumatic brain injury to some degree, we would have been somebody's breakfast. So we have evolved, the brain has evolved mechanisms with which to recover. So everybody has a mechanism to look at the pathophysiology of traumatic brain injury. Perhaps we need to look at the neurobiology response to traumatic brain injury and maybe help Mother Nature what it's trying to do. To try to take your target, your favorite target, and translate it into human conditions, into the ICU or to the emergency room, probably one of the best things we could come up with in terms of the laboratory is to try to determine if we've got an animal that is a responder versus a non-responder. And you can use that involves using non-invasive techniques that could be used in the intensive care unit. So if you've got for any type of reason, you've got an animal that doesn't respond well behaviorally to your drug, you may be able to look at the mechanism itself that you're trying to affect. And if it's not responding correctly, then you're wasting your time. You move on. And it helps individuals for clinical trials. And I would say one of the biggest deficit's the wrong word, one of the biggest challenges we've had is doing group comparisons. And within subject studies may be a more profitable way to actually look at outcomes. Finally, the human brain doesn't mature until well after 25 years of age. And that was discovered and described for many years ago in the 1960s and 1970s. And most of our head injuries in the United States and throughout the world are happening between 16 years of age and 25 years of age. And so we have a developing brain that also interacts with this. And these things need to be taken into account. It's, the ICU is a fascinating laboratory. And I think people like Claudia Robertson, Paul Vespa, and others have taken that opportunity to actually be able to utilize that in a way that makes a real big difference, much so that the emergency room could be used.
I want to follow up on that point about within subjects. This is a big issue in the human clinical situation because no two TBIs are alike, basically, in the ICU. And group mean statistics have not been particularly helpful to us over the years because we can't get to that predefined p-value that we're looking for. And so David Pansikowski, when he gives his talk later this afternoon, is going to show this a little bit, but this is what we're doing with our advanced neuroimaging work, is we have totally abandoned the concept of group mean statistics. And the analogy that we use is, if you take studies of orthopedic fractures and just compare fractures in this group to fractures in that group, it's not helpful. And in fact, if only 6% of all orthopedic fractures are radial bone fractures, and then you try to understand what you're doing for radial bone fractures in the context of the other 94% of the fractures that are out there, you'll never detect a group mean statistic for an intervention on radial bone fractures inside of all of orthopedic trauma.
Yeah.
So we have had to basically work with these folks from Carnegie Mellon, these statisticians, to develop novel math to do within subject analysis, and it's the only thing that crossed us over into meaningful quantitative assessment of human brain after TBI with neuroimaging. And I don't think we do that at the experimental level.
Yeah, I completely agree. The final thought is that we've heard some wonderful talks today, and all of us have written grants and shown our best work, and everything we've tried to do in the best way we possibly can. But our students, at least in my opinion, sometimes the students and fellows have forgotten that there are failures in the laboratory, and they shouldn't be afraid to do that. You shouldn't be afraid to try an experiment and it doesn't work out, or it doesn't come out the way you thought it did. Try to learn from it. Even though you may not be able to publish it, don't be afraid to try that. And the fear is always you're not going to be able to get funded, or you're going to be short on money. And that's a big threat, I think, to our field.
Excellent. Okay, we're going to move on to the...
One very quick closing comment, very quick. I want
I have
again
one
to
too.
associate which we're hearing today, the milder from the more severe end of the spectrum, the clinical trials. The mild injury, and we've heard this, I guess, from Dr. Carlson, we heard this from Dr. Satin, heard it from Audrey Lafreniere. Mild traumatic brain injury is a disorder of circuit disruption, and several of you in the room through advanced imaging have shown that. When we get into more we have to look at how that circuit is disrupted physiologically, anatomically, and how the brain rearranges itself, and how we can expedite that system. And I think that's an emerging field that we have to score upon. Severe injury, because of its complexity, which we heard, is a completely different animal and is multifaceted, as David has alluded to. But I think we cannot lose sight that we have to consider mild traumatic brain injury in a different context from severe. Yes, Ramon?
Very respectful.
Microphone?
Or disrespectful.
You can be disrespectful,
Disagree with
Ramon.
that.
It's okay.
I think mild traumatic brain injury is also heterogeneous and complex.
Yeah.
And I think
Well, it can be. That's correct.
I mean, the studies that have been done through Attract TBI and that Larry Latour has done at Bethesda,
Right, I'm not
with
arguing.
early imaging, I mean, it really is complex. There is axonal injury, that's correct, but it's more than just that, and not a insignificant number of them have focal features, obviously much smaller than the severes. But boy, I don't think there is a categorical difference between mild TBI and moderate to severe TBI. It's
Well,
a
those
spectrum.
patients with contusions are a different group, and people say it's a complicated mild TBI.
Yeah,
But
but
the
complicated
field
mild means that you can pick it up on a CT.
CT, that's
Right,
correct.
which is a technology that we've had available for a long time, but
Or a
what
biomarker.
about something that you can't pick up on a CT, but you can on an MR, right? Is that, what is that?
So there's an elephant in this room because you asked us to talk about the most important mechanistic feature.
Yes.
And the elephant in this room is that we've been talking about the heterogeneous presentation of traumatic brain injury, and our animal models are not so heterogeneous. And so what has emerged to some degree is a few efforts in combination therapies, where the idea is that I will address in studies parallel mechanisms and provide in the same therapeutic window either double guns for the same mechanism, or I'm looking at two different mechanisms at the same time, or for some of the funded combination therapies that just came to completion, it was serial. So first I'm going to do this, and then later in time I do this. All of these are considered combination therapies, and what was the emerging theme is that mechanism of interest that you study in a pure form and with a drug dosing studies that you do with timing and dose, it's all different when you have another agent on board, when you have the heterogeneous. So I actually don't even know how this community should be talking about addressing more than a single mechanism or one mechanism in more than one way in the setting of the heterogeneous traumatic brain injury, mild or severe.
Excellent comments and excellent discussion. We're going to move on to the next question. We heard today about very precise models of transgenic mice to study traumatic brain injury, to the other extreme of large animal models with concomitant hemorrhage and long-term ICU care. So the question that we had set aside ahead is, are there animal TBI models that reliably predict human disease in response to therapy? And if so, what are they?
Let me take a shot at that. We had a meeting, Dr. Pavlichak in Richmond,
25 years ago.
25 years ago,
Yes.
comparing and trying to answer that very question.
Right. Did we succeed?
And everybody has their favorite model that they've worked with, that they've characterized, and that they feel confident of, and that they can reproduce. And the quandary we had is that perhaps instead of trying to find a particular model that does everything, that matches the human condition, that we could take models that actually generate a particular component of the human condition. And it's the combination of those models of treatment that actually builds the model that you're interested in. So the straight answer to your question is no, there isn't one. The easier answer, or not easier, but the more complicated answer is that, and I contribute this quote to Dr. Pavlichak, it's a very complicated insult, and it's a very complicated organ that we're working on. And if you take these different models and combine them, you get a chance to get at certain aspects of the human condition. And to take a particular mechanism that you're going after, if you go across different models or different severities, you get even more enthusiastic that you're getting closer and closer to the human condition. But there are certain things that it can't answer. For example, traumatic brain injury doesn't happen to one person. It happens to the entire family. Well, we can have a model for that. In the military, we're trying to do resiliency and stuff like that for people. But I think that's the best answer that I've been able to come up with.
Yeah.
I agree.
I agree, and I think, again, at that report, which was sponsored by the NIDS, Dr. Kirsch Act, and it was widely published, widely disseminated, and summarily disregarded by the whole scientific community. It's very discouraging.
There were some fights in this meeting.
To write these consensus documents, but I think the bottom line, we said it's a complex disease. Obviously, you have to pick the models appropriate for the condition or for the disease entity you're studying, and then once you identify a potential therapy, it has to be screened across multiple models and higher order species, and that was the ending. And as I said, I thought it was an interesting document, but never saw the light of day.
But it's rare that that sort of expertise all lies within one lab, to have multiple types of injuries and multiple animal platforms, multiple outcome metrics. And so I think that's where team science actually comes in, and collaboration across labs, which is why I like your last question about how we can collaborate to accelerate.
I think either John or Ed can talk about this, but you've heard the Operation Brain Trauma Therapy a couple of times already. And it's basically an opportunity to go across different laboratories and different type of models and test drugs, compounds that seem to have benefit in well-done published publications on TBI. So we've been doing this several years now. How long have we been doing this? About 5 years. We've gone through a whole test, and hopefully some of these papers are going to come out soon, John. But
weeks.
what's really interesting, is that you can take one drug, and you can show that that drug has an effect on cognitive function after fluid percussion brain injury. That same drug may not affect cognitive outcome after controlled cortical impact injury, but has an effect on contusion volume. And you have another third model, a high-velocity missile, for example, and it has another effect on something else. So in each model, it's affecting different pathways, different pathophysiological conditions, and then, oh boy, in terms of the biomarker data, it's amazing the biomarker data across those different type of models, very, very different. So what we're learning is what we thought we knew and do know, that these models are quite different. And then you go to the pig, and John's working on trying to now move the positive effects up to the pig model, and that's another challenge. But it's very, very complicated. So I think we, as Dave said, no model's perfect, and we still do pretty much hypothesis-driven research, so we can actually ask that in a particular model because that pathophysiological mechanism is robust in that model.
Dalton, I can't resist one thing. I mean, the Operation Brain Trauma Therapy has been impressive, logistically that you guys have worked out, but one problem that has persisted, in my view, is that you are making the assumption, I realize it would be perhaps too laborious to do otherwise, but you're making the assumption that the dose that works in one model will also be the appropriate dose for the next model. And the severity of the different injuries are going to determine what the dose response is. So I need to be careful about that. So.
We have a question from the floor. Hassan?
I just want to maybe bring this issue back up and push the panel to make a comment on that. With the recent everyone has read the negative study with progesterone in traumatic brain injury, and if you look back at the preclinical data supporting that, it was all from rodent studies, mice, rats, with elegant mechanistic data. Multiple labs reproduced those findings, but when we started giving it in human subjects, the trial had to be stopped for futility. A lot of people got the drug with a waiver of informed consent, a drug that they didn't need, that didn't benefit them. So there's an ethical, moral issue, there's a financial issue. So obviously there's no one model that will answer all the questions. It just won't happen. But should there be a litmus test model, a translational model, once we've identified the science, and it works in multiple labs, and we know the mechanism, before we take it to a clinical trial, should there be a thumbs up or thumbs down kind of a model in a larger animal, non-human primate, swine, something? Because if we had done that with progesterone, maybe we could have saved tens of millions of dollars and hundreds of patients getting a drug that didn't help them.
However, I will say quickly, there will be a paper coming out very shortly from Don Stein where they reanalyzed the data and they determined that they used inappropriate dosing in that study. So,
again, goes to the same issue. Had we tested the dose in a more clinically relevant...
No, I agree. And I'd like to hear the panel's comment, yeah.
Done a better study.
Yeah, I don't know if you, I mean, it's heroic for people to go up the phylogenetic scale in traumatic brain injury models, including the mini-pig. And some of the best traumatic brain injury studies that I've seen, or that in the literature, came from Penn, when they can use non-human primates. Now in the state of California, that's where I'm from, if I tried to get an IRB to approve a mini-pig or a blast study, I wouldn't be able to get it approved. It's not possible. So that's why we've emphasized the importance of having some sort of biomarker, not necessarily to predict whether an outcome is going to be better, but whether the treatment of the animal that we're looking at actually affects the neurobiology. So it's very expensive. I mean, the people that do this work, and I get the privilege of reviewing some of their papers and stuff. I think it's wonderful, but it's really hard for people to do these large animal studies.
Your point is the crux of it all, frankly, and one of the fundamental problems is that the Glasgow outcome scale has nothing to do with anything that not one piece of data that has been discussed today has anything to do with the Glasgow outcome scale. And that's the only outcome measure that the FDA will accept. So there's no way to tie anything that any of you are doing in the laboratory to something that could be meaningfully tied to a Glasgow outcome scale. And why is that what we want to measure anyway? It doesn't make any sense. So before we actually have a litmus model, we actually need a litmus outcome measure that makes dramatically more clinical sense than the GOS.
So I'll just follow on that point. The other thing that we didn't hear about today is that, remember, all of these patients are being stratified for these clinical trials based upon a Glasgow coma score. And I didn't hear any of you guys talking about what the GCS of your mice and rats and pigs were in these studies, because you don't use GCS. We hear a lot of discussion about different sorts of lesions and types of pathology, which actually we have the ability clinically to look at a lot of these things. So, I mean, obviously, we just got to wrap our arms around the heterogeneity here and stop, looking the other way. Cancer's been the same thing. We didn't try to treat one group of cancer with one kind of drug, which is exactly what we've been doing here. I think what we have to do is we have to look for, I think Ramon likes to use the term endophenotypes, or, we need to stratify. The FDA calls it an enrichment study, but we have to find a subset of patients that look enough like the kind of great work that you guys are doing in the modeling here, that we have outcome measures that are similar to the outcome measures that you're using. So, as you pointed out, most of the things that were done with progesterone, they used a Morris-Watermaze test, which is a test of memory and learning. The GOSC is not a test of memory and learning, but we have a verbal learning task. We've got the RAE, we've got the CVLT. There's actually a number of things we do, and it turns out that the CVLT is actually a pretty sensitive measure for looking at some of these things. So, just to leave with some hope here, I think we already do have, one phenotype, if you will, within this whole mess that we actually know what it is. And I like to use this as an example. It's an epidural hematoma. I mean, pretty much anybody here that's gone to medical school, every emergency medicine physician, if you show them a picture, they'll say, oh yeah, that's an epidural hematoma. We understand the mechanism. It's usually a bony fracture that injures an artery, and there's a very targeted treatment, which is neurosurgery, which is we go take the bone off, we bipolar the artery, and patients, with a few things to adjust for their baseline characteristics, have a pretty good outcome. So I think that, John is on the right track talking about some of these disconnection syndromes that we see. We're certainly seeing with 3T MRI that we're finding a lot of these folks that are in this milder range that have these disruptions. I think this is the kind of thing that you guys can model, and when we figure out what is the best outcome measure for this, is this some sort of multitasking type thing? Is this in a network task that we need to do? Whatever we need to do that. I think we can tie that together and then go look for another little slice of the population, not try to fix the whole thing, because we're not going to fix the whole thing with a single drug. And I think we just need to recognize this and start working more closely together to find out what those subtypes are that we need to study.
But Jeff, then, Operation Brain Trauma says the criteria is that it has to be good, it has to be effective over a wide variety of injury models that may have diffuse or may have focal injury, and it means it has to be good at all things. which is really not what you're saying.
That's not what I'm saying. I mean, the whole purpose, and we're going to hear a little bit
So,
later.
so we may be throwing the bababy out with the bathwater.
Well,
There
I
may
mean,
be some that are good.
I really applaud OBTT. I mean, I think that what they're doing is they're actually now thinking translationally in a very serious way. And all I'm saying is that we need to refine this a little bit. I mean, one of the ideas of the TRACK TBI study is to really, in a very granular way, look at the natural history of the disease, trying to identify biomarkers or drug development tools that we can use, whether it's an imaging biomarker or proteomic biomarker, to stratify better than the GCS, and really going through a whole litany of these outcome measures that might be more sensitive than the GOSE. So I think that we're all sort of saying the same thing. OBTT is trying to take what we've already been doing and looking at this in, I think, in a very translational way. And all I would say is that as we begin to get a much better picture of what TBI really looks like, then I think we'll be able to refine and say, well, let's maybe go for this sort of pathology or this constellation of pathology. You guys can model it, and then we might have something that's translational with the kind of outcome measures, you know, like uractigraphy. I mean, that's a great thing. I mean, we couldn't do that 5 years ago. Now we can put Fitbits on people. So there's some things, but we have to be thinking translationally.
Ramon?
Yeah, so I think I want to object pretty strongly to this idea of a litmus test based on an animal model, because I think that's almost certainly going to throw out a lot of potentially very useful drugs. I think the animal models have their place, but if you look at the history of drug discovery, it's actually, you know, and that's not just in neurological disorders, it's most things. It's actually pretty disappointing, and I think I think what we've been doing in reality is skimping or being chintzy on the phase 2 stage, right? I think if there is a post-mortem to be done on the PROTECT and the synapse efforts for progesterone, is that, you know, it was really based only on 2 phase 2 studies, one of which included only 100 patients and only had 25 controls, and the other was done in China, and everybody's throwing out their arms and they don't know what the Chinese are doing, right? I think if there is something that we could have done better was not to do a pig study with progesterone, but rather to do some more phase 2 studies, and hopefully with some sophistication using biomarkers, hopefully we would have gotten the dose right or things like that. So I was a little concerned, but I'm not surprised that, I mean, Walter, over half of the NINDS money on TBI is spent in preclinical effort, and you know, you guys are great, but you know, I don't think that is probably the best investment. I think we've been skimping on at the phase 2 level, and you know, I mean, I think it's great to use pigs, it's great to use non-human primates, but these are very expensive studies. In fact, the cost of doing studies in pigs approaches the cost of doing studies in humans, and it's on a per subject basis. And
Ramon, I think your point is very well taken. We do need more sophisticated, larger phase 2 studies before going to phase 3. During the development of Terilizad, our head injury related phase 2 was really more about giving it to a relatively small group of head injury patients, proving it didn't kill them, and then going into big phase 3. So we need more detailed phase 2 studies. The other thing is I want to support Jeff and David concerning looking at a subgroup of traumatic brain injuries, and they've heard me say this before. If you look at epidural hematoma, then the epidural hematoma models are probably more relevant than the head injury models per se to determine that subgroup. Similarly, when we developed Terilizad, the preclinical data we had suggested that in patients with subarachnoid hemorrhage, this is a great drug. And indeed, we were able to prove in aneurysmal subarachnoid hemorrhage to at least to the point of getting approval in several countries for that Terilizad was useful. When we did the one only successful phase 3 head injury trial, the subgroup analysis showed that in those patients with traumatic subarachnoid hemorrhage, and primarily in male patients, there was something going on that could have been tracked down in subsequent trials. So we really have to be more specific. You know, I do a lot of work, as Dr. Dixon and others here, with the controlled cortical impact. But as I'm doing those studies, I'm thinking this is relevant to brain contusion. It is not relevant to diffuse TBI, in contrast to the central fluid percussion or a weight drop kinds of models. So I think we're on the right track, but we've got to, stay the course in this regard.
Okay, we're going to let Walter have the last word and then move on to the final question.
Good. So I wanted to give an encouraging word because everybody's been very kind of self-flogging here today. So, I mean, I think, we've had troubles for sure getting therapy to patients, but, we know so much more about head injury now than we did 20 years ago. It's just unbelievable. So I think we should be really proud of the work that's being done because we're learning more and more, and there's a value to the basic science piece. I mean, you heard discussions today about mechanisms that no one has thought about before, and I think that's the basic science part of this, is to keep plugging away at this. And the translational piece, we always have to be thinking about that, but because the translation doesn't work, doesn't really invalidate anything. It just means we have to be more clever as we move forward. So, and then the other thing so that's the first point. The second point is just an observation that that's somewhat interesting when you look out at what the companies are doing now. So, and then this is maybe a simple oversimplification, but a lot of the companies now are looking for a drug that hits a target. And then they're looking for a disease where that target might be important and where it can be tracked. They don't care whether or not, so if you had a target and head injury, they don't care whether it makes the pigs better or, walk around better or eat better. They just completely bypass that. And so if you can actually go to the human and test in the human against the target, they'll jump those steps. And so the example that's in the back of my head is this difumarate for multiple sclerosis. So in multiple sclerosis, the key event was that they had a biomarker of gadolinium-enhancing lesions. So there were no therapies that were really effective in multiple sclerosis. They found these gadolinium-enhancing lesions. They found that when they tested drugs, they suppressed these gadolinium-enhancing lesions, and that basically translated to decrease in number of exacerbations. And once they did that, it was like a highway opened up, and they just give drugs. Do they block the gadolinium-enhancing lesions? So and the last one, difumarate, which came out of Biogen, they didn't do any animal studies whatsoever, as far as I know. They just went right to human. They knew it was safe. They knew what they were looking for, and they went straight to humans, and they hit the jackpot with an oral anti-multiple sclerosis drug. So it's just the idea that one other way to jump the cliff is to try to work the human side more strongly to develop things that we think are relevant to outcome. People talk about cerebral edema today. Clearly people die from cerebral edema after head injury. It's not irrelevant. It may not explain everything about TBI outcome, but it's something you can measure, and it's something that if you have a drug, you can test it. Straight to human if you have a really good biomarker, but it requires a lot more work in people. We have really intense, rigorous work in animal models. Compared to what we know about people and head injury, it's, that's fantastic. We know very little. So that's the other side that I think we have to bring up.
All right. Excellent perspective. I'm getting the bell. In order to stay on schedule, we're going to skip the third question, and we're going to go right to the fourth question, which is how can laboratories work together in novel ways to accelerate discovery?
One of the things we're doing is developing the preclinical common data elements, and that's in Press Now, Journal of Trauma, and that was put forward by Ramona Hicks a few years ago, and I think a lot of us here worked on that in some degree. We've noticed reviewing the literature that there's a lot of variables that aren't reported in the literature, like the gender of the animals, age, the things you would think normally are reported, and this type of mechanism will help us compare our data. The data will be out there for other people to analyze, which is another issue of statistical analysis within labs. So I think using common data elements for preclinical models, I think, will help collaboration.
You know, I think that of all the fields of neuroscience, I think the field of neurotrauma, with regards to basic science laboratories, and even in clinical research, there's such a collegial approach to sharing data, calling people up, emailing them. Laboratories are open, they're not closed. In some fields of neuroscience, I've certainly, and I'm sure some of my colleagues can testify to this, they'll close them, where they're worried about being first, or they're worried about getting credit for something, or they're developing a company. In this particular field, it seems like the majority, and I may be wrong, but I think the majority of people that I know in the field of neurotrauma are focused on the problem. And if I can't, if I think that there's a mechanism that I think somebody else, like Dr. Dixon or Dr. Dietrich, could do better than I'd be more than happy to share that preliminary data. Or if I needed an engineer, you know, I don't have no qualms calling Dr. Margulies and saying, listen, I got this idea, I don't know how to do it, would you like to do it? And I think that happens a lot behind the scenes. Whether you're talking about formally, like in formal grant applications, that becomes difficult, not so much in terms of the sharing of data, but it's costly to have a subcontract, as in terms of just sharing. That's why a foundation like the Massey Foundation is so good for our field, because this is money that is not locked. This is money that can be used in a very novel way to bring a group of people together without having to worry about expenses. So I think that there are novel ways of working together. We've explored them a lot in our Neurotrauma Society and in our International Neurotrauma Society. But what I think is the most fun about this particular field, if I can be that crass, is that everybody gets along and everybody wants to make a difference. And it doesn't make any difference who gets the credit for it.
Well, I criticized just a moment ago the Operation Brain Trauma program based upon the dosing, but I got to say, the way that group has pulled off a logistical nightmare, is really impressive, and they've had a very good, detailed working relationship doing that. So I'll let them.
So Dalton, you brought up the spinal cord injury analysis where different groups got different outcomes. And so in my own experience with Tina Duhame for our NINDS U01 study looking at cyclosporine A in a preclinical trial, we really had to protocolize our, not just our injuries so that we were doing things the same way across two labs, but also in our assessments and then we had data analysis done commonly by a blinded individual. And that was really important. It's actually hard. So even when we have a 30-year relationship, but even that, it generated difficulties because not all labs do things quite the same way. And so I think you've got to have that trust and that friendship and the collegiality, but you also have to have some formal types of important conversations about exactly how to handle things and how to handle things when the outcomes aren't going exactly according to plan. So,
yeah, Miami and the group in California with Ozzie Stewart were the first groups to get a contract from NINDS to replicate the most exciting spinal cord data. And on the surface, we were very excited about it. As I got into it, we did 6 drugs, and as I got into it, it got more and more depressing because of the fact that a lot of the information was not in the papers. So you're exactly right, one-on-one on the phone with people in Italy and things like that. You know, again, this is what you said you did you do this? And all of a sudden, they start varying the story and things like that. But it got really depressing, and I learned something in that exercise, that it's very difficult, as you just said, to exactly replicate something. In fact, it's impossible. So is that the reason you couldn't replicate it, number one? Number two, and then, you know, talking to clinicians and the heterogeneity of the patient populations we're trying to target, even in spinal cord injury. You have Asia A, it's a very heterogeneous group. So all of a sudden it occurred to us that, well, we're going to have different types of models, and that would be more powerful, and that's the OBTT came up, and Pat Kohanek has led that program just unbelievably well. It's not the litmus test of what should go to clinical trials, by the way. It's basically to tell the scientific field that it looks like, you know, a particular compound may work in a different model, and from that you can take points and efficacy, and then maybe you can decide if it should go forward or not. But most of the drugs that we're testing aren't getting a lot of points. So right now we're still in that infancy stage, if you will.
Just a quick comment with the OBIT trials is we made a decision early on not to have everybody do the exact same outcomes.
Right. That's a
We
different way
said,
to do
do
it.
what you do best. Do your order maze the way you do it. We'll do it the way we do it, and we'll really compare effect sizes rather than numbers to numbers, and came up with a scoring system to do that.
I'll just make one comment, sort of digressing what was said, but getting back to the central question about how do we accelerate collaboration and cooperation. And this has been a theme of many foundation and philanthropic groups recently. I think many of us, Jeff and I, were at One Mind, where this was the overarching theme of that foundation. And I think, as Dave mentioned, we're a very cooperative community, but there is a reality of academics and grantsmanship. People cannot be fully compliant or fully sharing information routinely, because many people are worried about promotion and rank, promotion and tenure, and unfortunately our academic system sometimes does put some barriers and limitations on what we do. It's imperfect, but it is an impediment to some degree of extensive data sharing.
Yeah, I think, as Dave pointed out, I think we're a pretty friendly group of scientists, clinicians, and we talk and know each other very well and communicate freely. But in what we've been doing in the spinal cord field is having a series of workshops, and those workshops are centered around basically thought leaders thinking about what are we seeing in our patient populations, what are the degree of spontaneous recovery, what power do we need to actually test a compound in terms of preclinical arena, what is the critical data that everybody needs to have before we think they should go to a clinical trial in spinal cord injury, for example. So I think we're doing a lot of that in TBI already, but again, getting people together to have these thought discussions about specific points, you know, be it which is the best mechanism, what's the best model, things of that nature, and then learning from the clinical trials, the failures of clinical trials, and bring that back to the preclinical models and have a very thought discussion about what went wrong and what can we do better is great. But if you have a pharmaceutical company that is really pushing a particular compound, many times we're very disappointed when we see that compound go to a clinical trial without the preclinical data that should be supported, and that still happens to some degree.
Okay, thank you so much to our panel. This is going to end our first panel discussion in our morning session on laboratory science. I think it has been some great scientific presentations and great debate and insight from some of the thought leaders in the field, so thank you again. We're going to start with the first laboratory science question, and that is, what are the most important mechanistic questions related to traumatic brain injury that remain unanswered? And we'll start with volunteers.
I'll start. So I think everybody in this room has a favorite mechanism that they like. We all have our hand on a different part of the elephant. And to me, there's an overarching mechanistic question